Hello, everyone. I am Danielito, and I will be presenting the current progress of my master's project on the effects of drought stress on nodulation, plant growth, soil nitrogen availability, and nitrogen fixation in alfalfa under Dr. Malinda Thilakratna's supervision. Here is the outline of my presentation. First, a brief introduction on these topics, followed by the research objective, then methods, results, conclusion, my future activities, and acknowledgement. Alfalfa is known as the queen of forages for having the highest quality forage. It has one of the highest protein and overall nutritional content among forage legumes. It has a worldwide distribution originating in Iran, Central Asia, and Siberia. Due to its nutritional value, it is highly palatable for our domesticated ruminants and is commonly used as their animal feed. Because of these reasons, they are grown in approximately 30% of Canada's arable lands and is a popular legume species in sustainable agriculture to improve soil health and fertility by being a nitrogen fixer capable of fixing 78 to 222 kilograms per acre per year. Here we have a schematic diagram about nodulation and the biological nitrogen fixation. Plants release chemical signals called flavonoids that binds to nod genes called NOD-D in the rhizobium, which activates the release of nod factors, which are chemical signals to the plants to begin root hair deformation and initiate the eventual nodule formation. And this nodule unit houses the biological nitrogen fixation in the plants. Plants acquire nitrogen from the air, then transfers them into the nodules to convert them into assimilable forms through biological nitrogen fixation. However, this process is an energy-intensive process which they gain from the photosynthate products. And these highlighted parts are essential processes and organs for nodulating plants that when affected by abiotic and biotic stressors can significantly affect the plant growth, yield production, nodulation, and its benefit in soil health. Drought is a severe deleterious stress factor to plants worldwide, prompting to yield losses. And so plants have developed responses to help alleviate the effects of drought on them, namely drought escape, drought avoidance, and drought tolerance. However, under these responses, plants have to prioritize one process over the other, leading but not limited to the following: poor plant establishment, growth and yield reduction, reduced rate of photosynthesis. And in the soil, drought can affect the rhizobia abundance and survivability, which leads to reduction of nodule formation, and nitrogen's activity, which leads to poor nitrogen fixation of the plant. And so, I aim to evaluate the effects of drought stress on plant growth, plant physiological parameters, nodulation, nitrogen fixation, and soil nitrogen availability in alfalfa. I performed a greenhouse pot experiment. I pre-germinated some alfalfa seeds and seeded them into pots with 3 plants each. After a week, I inoculated them with the appropriate inoculum, followed by labeling with potassium nitrate after 2 weeks for the eventual isotope dilution method. And at flowering stage, I induced drought treatment via gravimetric methods, where they are maintained at 80% field capacity for well-watered, 40% field capacity for moderate drought, and 20% field capacity for severe drought by applying the appropriate moisture level every other day. Following 3 weeks, I measured the leaf chlorophyll content and photosynthesis using the PhotoSynQ tool and inserted plant growth simulator or PRS probes, which are left for a week. These are anion and cation resin membrane that facilitates the adsorption of ions in the soil with the help of moisture. And after a week, these probes are retrieved, plants are harvested, and soil samples are acquired. As shown here in the image in the upper left corner, there are single units and of nodules as well as clusters. I counted them both separately and then summed them up for the total nodule count. Panel A shows that single nodules decreased following drought, and the same trend can be seen in panel B for cluster count. And so the total number of nodules is significantly reduced under both drought treatment as depicted in panel C. The reduction in nodule number is likely due to the decreased survivability of rhizobia in the soil, or the amount of photosynthate being allocated for root growth, and nodule growth. Interestingly, in panel A, photosynthesis is not significantly affected under drought. The problem in measuring photosynthesis is that it is an instant measurement that can be better described by other factors such as leaf chlorophyll content as shown in panel B. Leaf chlorophyll content decreased under severe drought due to the yellowing of the leaves and onset of leaf senescence. Conversely, leaf chlorophyll content increased under moderate drought. This signifies that nitrogen accumulated in the leaves to drive water uptake and to form more chlorophyll units. The accumulation of nitrogen in the leaves sends a negative signal to the nodule to slow down or turn off nitrogen fixation to conserve the essential carbon that is being used up in the process. For other physiological parameters, panel A shows that quantum yield is not affected by drought. Quantum yield is the incoming light that is being transported to the photosystem 2. Panel B shows that non-photochemical quenching activity is increased under severe drought. This is due to the less photosynthetic unit, which are chlorophyll, that converts light into energy, and so they have to dissipate excess harmful energy to protect themselves. Here, panel A shows that shoot dry weight is significantly affected by drought stress. However, only at severe drought does root dry weight is significantly reduced. This is because of the allocation of dry matter into the roots during moderate drought as a response to maintain a relatively healthy root system, albeit a stunted root growth. Here we have other root phenotypic traits. Panel A shows that root length is significantly reduced under both drought treatment, while root surface area in panel B and root volume in panel C shows that these traits are greatly affected under severe drought. So we chose 2 carbon cycling enzymes to measure glucosidase in panel A, which is responsible for cellulose degradation, and cellulobiosidase in panel B, which is responsible for the glucose hydrolysis. It is observed that both enzyme activities are not significantly affected under drought. Next, we have phosphatase in panel A for phosphorus cycling and NAG for nitrogen cycling in panel B. Like carbon cycling enzymes before, phosphatase activity was not affected under drought, and the opposite is observed for NAG activity, which is significantly reduced under drought. To date, it is known that enzyme activity is highly variable from research to research. It is said that the duration and severity of drought does not affect enzyme activity entirely, but much so by the physicochemical properties of the soil, such as temperature, pH level, aridity, etc., followed by plant cover diversity and soil microbe community. Interestingly, soil available nitrogen increased under drought. Essential plant nitrogen forms, nitrate in panel A and ammonium in panel B, is measured to have increased under drought. It is common knowledge that nitrogen uptake diminishes during drought, but this results begs an interesting question: has alfalfa rhizome deposition increased during drought? Rhizome deposition is the translocation of organic compounds into the soil via the root system to allow plants to interact with the rhizosphere. It may have been increased due to the decline of the root structural integrity, increasing the release of these compounds. In conclusion, nodulation parameters decrease under drought stress. Plant photosynthesis and physiological parameters are reduced under drought stress. Leaf chlorophyll increased under moderate drought, but declined under severe drought stress. Plant growth and root morphological parameters are reduced under drought stress. There are no significant effect on carbon and phosphorus cycling enzymes, while nitrogen cycling enzyme activity was reduced under drought. And lastly, available soil nitrogen increased under drought stress. In the future, I will be performing N15 isotope dilution technique to measure symbiotic nitrogen fixation and microbiome analysis to observe the effects of drought on soil microbiome community. I would like to thank my funders and especially for these wonderful people for their guidance and support on this project. Thank you.